So our next speaker is Dr. Kavitha Raja. Dr. Kavitha Raja is a senior professor and head of the Department of Microbiology at Sree Tirunal Institute for Medical Sciences and Technology, Trivandrum. She is also faculty at Clinical Epidemiology Unit of Medical College, Trivandrum. Dr. Kavitha has held several important positions, including that of Medical Superintendent at Sree Tirunal Institute for Medical Sciences, Nodal Officer and Mentor for setting up the BSL-2 facility for RT-PCR across Kerala, Founder Secretary for Academy of Clinical Microbiologists, Member of Board of Studies, Health Sciences, Kerala University, Member of National Task Force for Assessment of BSL-3 and BSL-4 facilities by SERB DST. She was also editor of Journal of Academy of Clinical Microbiologists and is present Vice President of the Academy of Clinical Microbiologists. Her research interests relate to antibiotic usage and antimicrobial resistance, and she has authored several scientific publications. With this introduction, I would like to request Dr. Kavitha Raja to have her presentation on the topic bugs that produce drugs, antibiotic production, a biotechnology miracle. Over to you, ma'am.
Thank you for that nice introduction. I hope I am audible now.
Yes, yes.
Yeah. So actually, after listening to all these talks, all in-depth biotechnology, I am not a biotechnologist. I am only a clinical microbiologist who deals directly with patients and the antibiotics and what is the treatment for them. So I don't do any antibiotic basic science work, please understand that. But I will be presenting something about how antibiotics are produced. Shall I share my presentation?
Yes, ma'am. It's all equally important. We have to see all aspects of this research plans, no, ma'am. Yeah.
I took this topic because I'm very much interested in antibiotics, and what is happening in the world now is that a lot of resistance to antibiotics is also coming up. And the main advantage of antibiotics is that the older antibiotics are still as effective as they are, though they may not be effective for resistant bacteria. So that is a very important point to remember. Antibiotics don't get old and you just forget about them. Whichever antibiotic has been manufactured, they are biotechnology miracles and they have to be preserved for mankind. No antibiotic can be thrown away saying that it is old. So I hope that message will go through to everyone. This is a term that I came across, antibiosis. Am I audible to everyone? Am I audible?
Yes, yes.
Yeah.
But did you move the slide, ma'am?
Yeah, I moved it.
It's still with the first title slide.
Oh, I think I will go out and. If I move like this,
Yeah,
can you
yeah.
see?
Yes.
Yeah. Then I will go to make it bigger. I think I'll do like. What about this? Has it moved?
Yeah, it moved.
One arrow has come.
Yeah, but it's fine. It's fine.
Yeah. So antibiosis, this 19th century saw the quest for antibacterial substances. And that is before that, the 17th and 18th centuries, microbiology was progressing in that newer bacteria, newer viruses, etc., were being found out at a rapid rate. So they were all discovered by the beginning of the 19th century. And after that, we started, because just finding a bacteria is not enough. You have to find something to go against to kill it. So from that time onwards, people, yes, they started trying to look for antibacterial substances. And Waxman in 1941 coined this term to be life against life. So antibiotic means life against life. And later antibiotic specifically came to mean a substance produced by one form of life to kill another form of life. So this I want to make as the base of my presentation, because what I'm going to show is that all antibiotics are produced by microorganisms only against microorganisms. So this is a competition for survival in the microbial world. That is why they produce the antibiotics. So in the 19th century, let us see what happened. There were many observations. That is, fungi were capable. One scientist brought up a paper on fungi were capable of inhibiting bacterial growth in certain culture media. So they don't know what to do with that, but they observed all these things. Antagonism between the fungus, Penicillium glaucum, and bacteria by animal experiments. Another paper. Then substance produced by Bacillus fluorescens inhibited Staphylococcus pyogenes growth. That came up in 1887. So people were observing a lot of things. And what are these antimicrobials? They are chemicals produced by microbes that kill other microbes in the fight for survival. And in the present era, these antibiotics are specifically against bacteria. And others are called antifungals, antivirals, antiparasitic agents. And for antimicrobials to be effective, the effect must be systemic after being absorbed either through the alimentary tract or by injection, reaching the infected area by way of the bloodstream. So each of these points are important. All other agents designed to kill microbes applied topically are merely antiseptics. So that is what, so now we don't say antibiotic, we say antimicrobials, and the resistance is called AMR or antimicrobial resistance. Then we will start with Fleming and the mold of called Penicillium notatum. Everyone knows about Fleming and how he discovered Penicillium notatum and its effect. He went on a holiday, and after coming back, he found that on his plate, his petri dish where Staphylococcus was growing, there was a fungus growing, and around the fungus there was an area of inhibition, which means the Staphylococcus was not growing up to the edge of the fungus. It was growing a little away from the fungus, which means that the fungus was preventing Staphylococcus growth. And then though it was a serendipitous, that is, he just found a plate like that. Somebody else could have just thrown the plate out, saying that there is a fungus inside. But he found that plate very interesting, and he took the penicillium and started doing study on that fungus. It proved extremely effective, he found, even at low concentrations, preventing Staphylococcus growth even when diluted 800 times, and was less toxic than the disinfectants used at that time. Now, how the antibiotic evolution of the
did you move the slide? What slide?
Yeah. Okay, I'll go back to this. I am on this slide now. Did you see
Second
this slide?
Second slide.
This slide.
Okay.
Then the third slide, observations. Yeah. So this one you didn't see. I think I'll keep it like this. Is it okay if I keep it like this?
Yeah, that will be nice. Yeah.
I'll keep like this. Otherwise, it's not moving. Then
yeah.
I'll try once more.
Okay.
Now I'm on this slide. Antimicrobials.
It is moving.
Yeah. So that is the next one. So the chemicals produced by microbes that kill other microbes in the fight for survival. They all have to survive in the media. And now we are calling antibiotics, antifungals, antiviral, and antiparasitic agents separately. Then coming to Fleming. This is Fleming. You might not have seen the slide. This is Fleming, and he is just discovered Penicillium notatum, and he found that it's effective even at very low concentrations, preventing the Staphylococcus. So you must remember that till now, lot of observations were made. So these were the observations made before Fleming. So actually he was not doing anything very new. He was just standing on the shoulder of giants, so to say, that he found that there were other articles like this where substances are produced. So here he said that this is very interesting fungus. But he could not do anything more than that. He could just show that they are infective. Then what happened was he went into, can you see the next slide now? Is it visible?
Yes, now it is the sixth slide.
Yeah. Okay. So the evolution of manufacture of antibiotics, I'll say one word about this, then we'll go back to Fleming. So earlier antibiotics were discovered by two means. That is from fungi, for example, penicillin. From bacteria, streptomyces, actinomyces, all produce antibiotics. Then the next thing that came up was advances in biochemistry. Synthetic derivatives of these antibiotics, that is penicillin, streptomycin, erythromycin, they advances in biochemistry caused better structures, more effective molecules came up. So they were synthetic derivatives of the natural form. So there are two types of antibiotics, the natural ones and the synthetic ones. Then came the advances in biotechnology, where it was not just synthetic in the chemistry lab, but biotechnology, where chromosomal alterations and use of fungi and bacteria in mass production systems came up. So with this background, that is, first it comes from the base antibiotic, always comes from a microbe, fungi or bacteria. Then there are two ways you can modify those and bring up other molecules. One is through biochemistry, other is through biotechnology. Now, is this visible? This slide, next slide.
Yes, ma'am.
Yeah. So, how do you get a new antibiotic? So I said that you can get it from fungi and you can get it from bacteria and then do some chemistry on it. But how do you get a new one? We already have lot of antibiotics and we have lot of synthetic ones also. But we don't want all these. They are all resistant. Then what do you do? How does a scientist go about to get a new antibiotic? One is a soil survey. That is, you take the soil and you try to culture it and grow bacteria that are actually commensals or saprotrophs, that is growing outside the human body in the environment, are called saprotrophs. Generally, they be non-pathogenic for human beings. So we can try to find antibiotics in such bacteria which are not dangerous for human beings. So the soil survey can be done. Bacteria are grown and these bacteria are taken and action against pathogenic bacteria can be tried. So that is one method. Another method is the fungi, that is molds growing in different places. So you get on your bread, you will get some fungus growing. Or one interesting place was sausage, cured sausage. That is on the surface of the sausage, some molds grow, and it increases the some nice taste, tangy taste comes for the sausage. So that also was taken up. People thought why people are not getting any disease even if there is a mold growing on the sausage. So that mold was tested and it was found that it has antibiotic properties. So these are the two ways you can get a new one. And what are the properties looked for? It should exert the antimicrobic action in high dilution, whatever dilution, because you have to put it into a human body. You cannot put a concentrated antibiotic and cause toxic effects inside the human body. So it should be effective in high dilution. It should be harmless to human tissues. It should be very stable. It should have a slow rate of excretion. Shouldn't be quickly excreted without any action. Diffusibility, yes. It should reach different areas of the human body, like the interior of the lungs, the brain, and the bone. So all this, so if you say that you have a new antibiotic with you, it's not enough that you show that it will kill some bacteria. you should also show all these properties for saying that this is a new antibiotic and it can be used on the human body. So the next slide shows source of natural antibiotics. So I'll just examples of the natural antibiotics. Can you see the slide? Example of the natural antibiotics. Penicillin.
Yes.
Yeah. So penicillin from Penicillium notatum, streptomycin from Streptomyces griseus, erythromycin from Streptomyces erythraeus, vancomycin from Streptomyces orientalis, gentamicin from Micromonospora purpurate, cephalosporins from Cephalosporium species, polymyxin from Bacillus polymyxa. So these are just a few examples. Now we'll go back to our story of penicillin, and this is a very classic example of how the over the years how the technology developed. Even now, penicillin production is being made better and better, easier and easier, and more amount of the drug. So the drug is still extremely useful for human beings. Now, after Fleming, there were two people called Florey and Chain, who in 1940 devised a method to extract it out. So Fleming was using it as a fungus itself, but Florey and Chain extracted the actual penicillin, that biochemical that content from the fungus for mass production. And that came as an article as Chain et al. 1940. And because of this, in 1945, Fleming, Florey, and Chain got a Nobel Prize. And this molecule, penicillin, saved hundreds of lives in World War II. And actually, it was World War II that fueled the quest for antibiotics, and especially to produce better and better forms of penicillin. So when I just said like this, that they extracted it in fungus and there was mass production. Is it so simple? So this is my next slide. Let me just put it into the whole screen. Is it visible now?
Yes, ma'am.
Yeah. So the Fleming, is it? I mean, it's a full screen now. So Fleming.
Screen for us, but we can see.
Yeah. So I have written the names of the people on the left side. That is Fleming. He had only a crude method. He used the fungus and a bit of the extract he used to maintain pure cultures. He did not go any further. But Florey and Chain, with some commercially oriented people like Heatley, Abraham, etc., they decided that this has to be properly utilized. So they went for filtration, extraction, purification, but they remained with Penicillium notatum. And they tested it on, so the first testing occurred on mice. And when it became successful in mice, they used it on one human being. He had an injury to his face, and there was an infection there on his face. It's very dangerous when you have an infection on the face, because there are lots of arteries and veins there, and quickly you will go in for sepsis. So they gave this person penicillin. I think they must have made the extract and put it, topical application must have been done, because IV, I don't think they would have done. They would have put it topically on that person's face, and he was actually getting better. But there was a tragedy at that time that they ran out of the penicillin. Whatever they did with the extraction and purification and producing, it was a big, tedious process, and only little bit of penicillin was coming out of it. So this poor man, though he was slightly getting better with that, and they demonstrated it, they ran out of penicillin and it could not be continued. Treatment could not be continued and he died of sepsis, staphylococcal sepsis. But they were happy that at least it showed some effect on the human being. Then this was taken to Illinois, to US, and in the Northern Regional Research Lab during just before World War II, the US government said that this antibiotic will most probably become useful for soldiers because they will have infected wounds. So they were instructed, the Northern Regional Research Lab was instructed to produce as much penicillin as possible. So what they did was they added lactose as a growth medium and corn liquor also they added for the fermentation process to be increased. So, and then they added a compound called phenyl acetic acid, which is a precursor for penicillin. So all these things they added. So they were thought if it runs out of phenyl acetic acid, then the penicillin production will come down. So all these things they tried, and they also changed the penicillium. They took another penicillium species, Chrysogenum, which they found produced more penicillin than notatum. And this was from a moldy cantaloupe, that is a sweet melon. And this new penicillium, Chrysogenum, was used, and then they found that they could increase the amount of penicillin to a great degree. And this was then later on, this has become the largest produced antibiotic in the world today. Even now, penicillin is the largest produced antibiotic in the world. Now, no other antibiotic is produced in such large amounts. So why are we producing such a lot of penicillin? Let me see. Today, the demand for penicillin is more than the supply. Why? Because 30% is used for humans as penicillin, 60% is used for making synthetic penicillins like penicillin B, G with other properties, benzathine penicillin, and 10% is also used in the animal industry, especially food industry, to keep the animal healthy and all. So actually it's a bit of a misuse, but this is why the demand for penicillin is still more than the supply. So unless I go to the main screen, I can't show my animations. So is this visible now?
Yes, ma'am.
Yeah. So how is it done actually? So Penicillium chrysogenum, we will start with that. And now it is called Penicillium rubens. It was made to produce more and more and more of penicillin. How? This was done using UV rays and X-rays to mutate the Penicillium rubens. And this method was called classic strain improvement. Basically, there were some mutations inside that fungus. Penicillium, then this fungus, because it was done in the regional research lab in Illinois, it was given the name NRRL 1951 strain. And then what was the mutation that occurred there? As I told you, phenyl acetic acid was a precursor for penicillin. And so what they found was this mutation. So the mutation, what is the function of that mutation? They tried to look at that. And they found that this is a mutation that prevented the destruction of phenyl acetic acid. So inside a bacterial cell, when some compound increases in amount, naturally that bacteria doesn't want a lot of phenyl acetic acid hanging around. So what does the bacteria do? It will destroy it only. It will make only as much as it needs. But human beings want a lot of it. And this mutation using the X-ray and UV ray prevented the destruction of phenyl acetic acid. So that the fungus was converted into a fungus that was mainly producing lot of penicillin and no other function. And then the University of Wisconsin also took this up, and they found that if you inhibit other functions of the fungus, that is of the fungal cell, if you inhibit other functions, other metabolic activities, more of penicillin, more of penicillin production will occur. So what they did was a pigment. There is no need of it. This pigment was not useful to us. So that was also inhibited. And that was called the strain of the Wisconsin. Number is 541255. After that, they found that keeping it in the air and making it and producing penicillin was less efficient than submerged culture. So you go on trying different methods, and one method was submerged culture, and aeration increased the yield. And this was a revolutionary technology at that point of time in the 1945, 46, 47 time, that is during the World War II at the beginning stages. This became revolutionized, and this was then taken up by the companies. The multinational companies took this up, and of course, even now the same name comes up, isn't it? The vaccine also. So Pfizer started off with this miracle, that is on March 1, 1944. Pfizer opened the first commercial plant using submerged cultures for penicillin production. That was the only antibiotic available at that point of time, and it seemed to be a big miracle, because the moment you give it to a patient with severe infection, within 24 hours the patient improves and there is no infection. In about 2 to 3 days completely it is resolved. So Pfizer found that it was useful and it will be very profitable also. So which are the genes in? Okay. I think I have to go the other way only. Yeah.
No, ma'am, it's fine. But we have some time limitations. So
okay, because I'm doing this. Yeah.
Yeah. Okay.
So what will be the time? 10 minutes? 10 minutes?
Yeah, 10 minutes.
10 minutes. Okay.
Sure.
So this is the one. So this is the genes involved. That is, so we go to the gene level. That is PCBC gene, PEND. So you can see it here. This is the Wisconsin strain. This is the Fleming strain. This is another strain. So many strains came up, producing better and better penicillin. And the commercial production, you can see here. This is the old way how it was produced. 1944, the Brooklyn Pfizer deep fermentation technique using this, and then the packaging of the penicillin vials in the plant. And this is the storage vial for penicillin. So this is all of great importance, historical importance. And this is the big plant only for penicillin by Pfizer. But later on in 2008, they moved to another place and this plant was demolished. But you can see the importance of producing this antibiotic. Just wanted to show that. Then came the era of genomics, chromosomal rearrangements, amplification of gene clusters, point mutations, and the fungus itself. We found that there is a heterothallic, that is, there is sexual reproduction. All this changed the methods for production. The modern era, genetic engineering. So this gene locus controls sexual mating in P. rubens. And this P. rubens, this strain P2NID18 retains the ability to sexually cross, opens a possibility of using. So these are the biotechnology things that came up with penicillin later on. So you saw how it evolved over the years. Now this one I think I will have to show in the big screen only. There's no need of going into each and every sentence here. Just wanted to show you that over the years, even up to 2020, that is the message I want to give. The genome sequencing is being done, genome editing using CRISPR technology. So this is how biotechnology is advancing the production of penicillin. So better penicillins and large amounts of penicillin are being produced. So even up to 2020, the research into penicillin is still going on right from the 20th century, that is from 1944 onwards. So that is the thing. So don't say that antibiotics are old. all antibiotics, old or new, have their use, and they are very efficient now also. I'll just quickly go through the cephalosporins. Also, in the same way, because it's a very interesting story. There was a professor called Broadsu who questioned why typhoid fever was less virulent in his city than elsewhere. And he found that he took samples of water from the area where many youngsters were swimming, precisely at the site where the city's sewer system drained into the sea. Still, they were healthy and nothing happened to them. So why was that? He took the sample of water and cultured in his lab, and it was so suddenly he isolated a fungus so easy. Don't think that it is as easy now, but he isolated a fungus that produced an infective substance against gram-negative bacteria. And what was that fungus? That was Cephalosporium acrimonium, and that is producing the cephalosporins. Were developed from Cephalosporin C, a natural product of Cephalosporin acrimonium, and cephomycins are related drugs from streptomyces. This is a bacteria. Cephalosporin acrimonium is a fungus. So once you get a product, then you can also try to get it from other bacteria and fungi. And this is the difference. See, the cephalosporin acrimonium. So the common area for penicillin and cephalosporin is this area. And from here it becomes penicillin. This is the pathway for penicillin. And penicillin has this beta-lactam ring, this square thing you see here, with an attached thiazolidine ring, which has only 1, 2, 3, 1, 2, 3, 4. Only the 4 things in this way. It's a diamond shaped ring. But when you go for cephalosporin, it has 1, 2, 3, 4, 5 things, and 6 bases will be there. So see the difference between this. This is a pentagon. This is having 6 points. So this is the difference between penicillin. So what does it do? This acrimonium extends the biosynthetic pathway for penicillin and goes on to produce cephalosporin C. So cephalosporin is related to penicillin, but this fungus has learned how to survive better and produces a later form of penicillin. So this is how it has produced another form. And the production, what was done is that UV rays induce again the same story, mutations, and they went on. This Broadsu strain has been changed now, and it has become the CW19 strain, and this produces 3 times more cephalosporin than the Broadsu strain. So finally, now we have come to the genetic engineering phase for all antibiotics. So for any antibiotic, what is done now is the genes responsible will be identified. Multiple copies of the gene can be made. And after that, you decide what you have to do. You have to overexpress the gene somewhere. So CEP gene is overexpressed, and molecular biology and its practical counterpart, genetic engineering. This has been incorporated into the strain improvement programs of most pharmaceutical companies. And the new cephalosporins, not only genetic engineering, we have a lot of chemical synthesis also. The newer ones, first, second, third, and fourth generation cephalosporins, by changing the molecule in different ways. This is another way you can improve upon antibiotics. Then we'll come to the next antibiotic, just one more I have. That is the gentamicin, which is a little different from the other beta-lactam antibiotics. This is from a micromonospora echinospora. This is our purpurate, gentamicin. This is the soil you find in the soil, aquatic environments. And why is it MIC, not MY? Because it is micromonospora. This MYC we have added here. So ending of mycin indicates micromonospora as origin, while ending of MYCIN is streptomyces, erythromycin, etc. And this is the micromonospora species. You can see that it is purpurine, means it is reddish color. So this is not a fungus and it is not a bacteria. It is something in between bacteria and fungus. A bacteria that has grown something like the hyphae of the fungus with branching, and but it retains the spores of bacteria. So this, and it grows on ordinary bacterial media, produces a pigment. Micromonospora species. All these are non-pathogenic to human beings. And you can see what all antibiotics are produced from this. From the micromonospora species, aminoglycosides, quinones, alkaloids, ansamycins, peptides. So one species itself, we have started finding there are so many antibiotics can be produced from that. And this is how the gentamicin is produced. This is synthesized. One small problem was there. It was synthesized inside the bacterial cell and it has to be extracted. So that is the gentamicin structure. Aminoglycosides, just to give you a clinical touch for that, gentamicin, amikacin, netilmicin. So many things have come out from that original molecule that I showed you, gentamicin. They have broad spectrum antibacterial activity against gram-positive and gram-negative bacteria, mycobacteria, and protozoa also. Their mode of action is they will prevent protein synthesis inside the bacteria. But they will not affect human beings because human beings have a different ribosome. This is 16S ribosome. So it's a different ribosome for human beings. So it doesn't affect human cells. And does India have a plant for antibiotics? Yes, it has. There is a Hindustan Antibiotics Limited at Pimpri, Pune. In between, these are closed down and all that. But now again, it has come up, and this is the plant at Pune. Do you have any research for antibiotics? This is the research is being done in the National Institutes of Pharmacological Education Research, NIPR. These are, there are lots of these institutions, Mohali, Hyderabad, Ahmedabad, Nagpur, Guwahati, Kolkata. So if anybody is interested, they can follow, they can try to find new antibiotics, do research in these institutions. What are the problems in the industry? Basically, the first problem is, of course, antimicrobial resistance, so that whatever you have is of no use. Scarcity, like I told about penicillin, the growing demand is there and supply is less. New antibiotics in the form of absolutely new. The new ones that are coming up are all synthetic. That is, the chemical is little changed to produce new. But absolutely new antibiotics are not there now. No new antibiotics. Then old antibiotics, which were there earlier, are being recycled because of the mounting antimicrobial resistance, so that there are adverse effects. So there's a real need to find out new antibiotics. But industrial support is diminishing because nobody is interested in producing new antibiotics. It caters only to a small group of people with infections. So companies are also not interested in producing new antibiotics. Then the mega companies, lobbying by mega companies, what they do is when they produce a new antibiotic, older antibiotics they try to suppress. They reduce the supply of the older antibiotics, and that can also affect, in turn, cause more antimicrobial resistance. So these are the problems in the industry. And as conclusion, I would like to say that antimicrobials are nature's secrets for survival of the species. Human beings have harnessed these secrets of microbes and are using them for their own survival. Antimicrobials have increased human life, a span by about 30 more years. So it is really important. fast advances in biotechnology are changing the way we view infectious diseases. But the final word is always prevention. Prevention is always better than cure.
Thank you, ma'am. The talk is open for discussion. We can take one or two questions because of the time constraints. Just one quick question from the audience. Ma'am,
Good
yeah,
afternoon, ma'am.
yes.
Yeah.
Thank you, ma'am, for a wonderful presentation. This is an interesting topic, of course. My question is related to the current research that is published by Lancet, which is setting an indication that there is an increase in antibiotic resistance, which is on a global scale. Even if we see E. coli, Staphylococcus aureus, or Pseudomonas, they could be the deadliest organism in coming future. So how this problem can be targeted, because there is an overuse of antibiotics.
Yes, as I said, prevention is always better than cure, and antimicrobial resistance is a big problem now. But what I have seen from my experience is that mostly this antimicrobial resistance occurs in large referral super specialty hospitals with intensive care units. So inside the intensive care unit, doctors are very worried and they will just give all kinds of antibiotics. For one patient, they will give 2 or 3 antibiotics. And as you know, as biotechnology students, you should know that these microbes, they have to survive. So they are actually biotechnology experts and they will change their chromosomes quickly and they will adapt to the environment. And what happens is that they will become resistant to whatever antibiotics. So the penicillin that was discovered, the microbe quickly produced beta-lactamases. That is, beta-lactam is the antibiotic. Beta-lactam is killed or neutralized by the antibiotic that is released, will be neutralized by an enzyme called beta-lactamase that is released from the microorganism, so that the bacteria is not killed. So just like that, they are also factories producing some drugs for them, which will prevent their death. So if you increase the amount of antibiotics in the environment, especially inside the hospitals. Outside the hospital, it is not that bad, but inside the hospital, it is always increasing. Another thing is outside the hospital, there is use in animals, there is use in aquatic culture, there is um, what do you call it, the effluence that is coming out from hospitals and from other places where animals are being looked after, or from all those places, the effluents, they may also contain antibiotics. And the antibiotics will get into the environment. So all this can add on to the AMR. And as I think biotechnologists, we have to find new antibiotics and we also have to find ways how to neutralize these products that are produced by bacteria. So that is the challenge for the future.
Yes, ma'am. Thank you. Thank you very much.
Thank you, ma'am, for the wonderful talk. Due to time constraints, I would request the audience if there are any questions to email ma'am for more clarifications.